Good afternoon, everyone. A heartfelt welcome to all participants joining us from across India and around the globe. We, the students of People's University, are proud to present this special webinar designed and driven entirely by the students for the students. On behalf of our organizing team at the Center for Scientific Research and Development, People's University, Bhopal, India, I warmly welcome you to the 8th session of the BioConnect webinar series. I am Chehak Soni and I am proud to tell you that I am a student of BSc Biotechnology and today's MC. It is truly a great honor to host this insightful event. Today's webinar focuses on a timely and thought-provoking theme, biotech tools of sustainable topical crop production. We are deeply honored to welcome the eminent guests and leaders joining us today. A very special welcome to Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, Professor Dr. Jennifer Ann Krishna, our distinguished speaker, Professor Dr. Subhash Bhore, our mentor and head of the Center for Scientific Research and Development at People's University, and Mr. Mohammad Arshan, Chairperson of the Organizing Committee of this program. Now I invite the Chairperson of the Organizing Committee, Mr. Mohammad Arshan, to say a few words. Arshan, over to you.
Thank you, Chehak. Good afternoon, everyone. Our esteemed guest speaker, Professor Dr. Jennifer Ann Krishna, our Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, our respected mentor, Professor Dr. Subhash Bhore, distinguished dignitaries, respected faculty members, and my dear fellow students. I extend a very warm welcome to you all to this webinar session. My name is Mohammad Arsan. I am an MSc Microbiology student at CSRD People's University, and I am truly honored to serve as a chairperson of the organizing committee of this program. On behalf of the organizing committee, I am delighted to welcome you all to this thought-provoking webinar titled Biotech Tools for Sustainable Topical Crop Production, case study from University Malaya. Agriculture today faces multiple challenges ranging from climate change and soil degradation to pest infestation and reduced crop productivity. To ensure food security and sustainability, biotechnology provides a powerful toolbox of modern solutions from molecular breeding, genetic engineering to biofertilizers, biopesticides, and precision agriculture technique. Case study from University Malaya have shown how these innovative tools can be applied to tropical crop system, ensuring resilience, productivity, and sustainability in one of the world's most diverse agriculture region. On behalf of the organizing team, I deeply honored to welcome our distinguished guest speaker, Professor Dr. Jennifer, whose expertise and pioneering work in crop biotechnology continue to inspire students and researchers worldwide. We are confident that her valuable insights and case study will enrich our understanding of how biotechnology can transform tropical agriculture for a sustainable future. With these words, I thank you all once again for being with us today. Let us look forward to an inspiring and insightful session ahead. Now I hand it over to MC. Ms. Jack, over to you.
Thank you so much, Mr. Mohammed Ashan, for your encouraging words and welcoming remarks. Fellow students and esteemed participants, it is now time to begin the core session. To take it further, I hand over the session to our moderator, Ms. Devyani Dan. Devyani, over to you.
Thank you, Chahal. Hello, everyone. Very warm welcome to all participants joining us from across India and around the globe. I am Divyani Dali, and I am proud to tell you that I am a MS Technology student at the Center for Scientific Research and Development, People's University, Bhopal, India. It is my privilege to serve as moderator for today's insightful session organized under the BioConnect webinar series, an appreciable initiative of the CSRD People's University. Today's session focuses on the topic of significant relevance in today's fast evolving scientific landscape. Today's talk will focus on biotech tools for sustainable topical crop production. It is my privilege to introduce today's distinguished speaker, Professor Jennifer Ann Hari Krishnan, Professor of Microbiology and Molecular Genetics, Institute of Biological Sciences, Faculty of Science, and Center for Science Research in Biotechnology for Agriculture, Sibar, University, Malaya, Malaysia. Professor Jennifer Ann Hari Krishna, originally from the UK, is a distinguished scientist in the field of plant biotechnology. She completed her BSc in microbiology at the University of Surrey before earning a PhD in molecular genetics from Cranfield University, where she received the Chancellor's Gold Medal for being the most outstanding graduate student. After a postdoctoral fellowship at the University of California, San Francisco, UCSF, she moved to Malaysia and has since contributed extensively to agricultural biotechnology through positions at Trop Bioresearcher, University Putra, Malaysia, the Malaysian University of Science and Technology, and currently University Malaya. At University Malaya, she served as the director of SIVAR and as a professor of the Faculty of Science, where work focuses on advancing biotechnology for sustainable agriculture. She also maintains international collaborations as an honorary visiting fellow at the University of Leicester, UK, and Gadjah Mada University, Indonesia. Professor Jennifer's career reflects her strong commitment to using cutting-edge biotechnology to address agricultural challenges, particularly in topical crop production. Today, Professor Jennifer will speak on biotech tools for sustainable topical crop production, highlighting innovative approaches such as genetic engineering, molecular markers, and bio-based solutions that enhance crop resilience, reduce chemical inputs, and support environmentally friendly agriculture. With this very brief introduction, it is my honor to invite Professor Jennifer to deliver her talk on biotech tools for sustainable topical crop production. Professor Jennifer, over to you.
Thank you. And thank you very much for the warm welcome. And I also want to congratulate all of you for this initiative. It is wonderful to see students running this type of event. And it bodes well for your future that you're interested in science and networking, especially with scientists from around the world. Let me just go to full screen. And can I check, is that on full screen for everybody?
Yes, yes, we can see.
And can you see, if I move the arrow, can you see the arrow?
Yes, yes,
Okay.
yes, we can see.
Okay, so I can use that as a pointer. That's great. Okay, so I'm just going to give you some small examples from my own work. And when I say my own work, of course, it's lots of students and colleagues helping. It's not just me. And but it's been really fun. And I've been working at University Malaya now for almost 20 years. And these are some of the projects that we are most excited about. So first of all, why do we choose to work on crop production and why is sustainability important? So I think never more than today, the world is facing a lot of challenges. If we even forget about the geopolitical ones, we have climate changes, we have diseases, and these can reduce the quantity, the quality, as well as availability and affordability of food. So what can we as scientists offer to help? And this is what I think is very important. How can we contribute not just to our own publications, but to try to make a difference for our planet? And one of the things that excites me about biology is the amazing number of solutions that it already has solved. So we have to discover which of these solutions that are already there in biology can be adapted and applied. And that is really what biotechnology is. It's not really inventing something new, it's seeing what is there and how we can use it. So today I'm going to take you through two biotech solutions. One is actually a very old, not really cutting edge, but it's moving into cutting edge these days, and that is using microbes to alleviate stress. And the second is something more current, which is the use of small RNAs, like RNA sprays and microRNAs, to reduce the reliance on traditional agrochemicals and to go back to something that may be less harmful to the environment. So this is the reality you as young people face even more than me and your professors. We are getting older now, but you have many more years to live on this planet. And you can see clearly that it's getting hotter, and that is definitely going to impact the performance of our crops. And it's also getting wetter. And on the right-hand side here, I'm just showing you some projections for Malaysia, where I am, around Kuala Lumpur. And the coastline, you can see currently where it is. It touches where the blue is. But that part in the sort of reddy pinkish color could be completely underwater by the end of the century. And that is what is predicted with sea level rising. And it doesn't look so big on the whole map, but this is a huge area where agriculture is conducted. So it's going to have a big impact. And around the world, you can see many countries, including India, are going to be very severely affected. And we already see that there is much flooding in our countries, India and Malaysia, over the last few years. But one of the things that comes with flooding is salinity, because seawater encroaches into groundwater or over land when it floods. And also it can impact through water scarcity. When the water falls, we have plenty of water. When it goes away, we need to irrigate, and that can reduce the quality of the water and result in buildup of salts. So salinity is also a tremendous problem that many countries, India and Malaysia among them, are facing. And we can also see changes in biotic stresses, pests and diseases, through the changes in climate, because they change the weather patterns, and that can change how the vectors and even some spores can be transferred across tremendous distances. So we can get new diseases that were not in our part of the world moving into our part of the world as a result of climate change. And this is really a great concern, because it could happen very quickly, and suddenly we're dealing with a new challenge. So I'm going to go to our first story, and I work on bananas. In fact, I was in Indonesia last week with people from all over Southeast Asia who work on bananas. So there's a lot of us, we call ourselves the banana bunch, because we're all interested in bananas. They're a very important food security item in many countries. They're highly consumed as a fruit, but in parts of Africa, they're actually used as a starch source as well, so not just as a dessert. And it's a very highly nutritious food. It's actually very productive per area land that is used. But like most crops, it's susceptible to water stress, especially drought, flood, and salinity. And it's susceptible to many diseases. The most well-known is Fusarium wilt, but I was learning last week that across Asia, we also have many other disease challenges for banana. So we wanted to look at what happens with sea salt flooding. So this is in line with my earlier statements that we can expect Malaysia to be affected by seawater rising and more seawater flooding in the future. So what impact does that have on bananas? So actually, it's not good. Here we can see a summary that 20% seawater even is quite detrimental and will dramatically reduce the yields. By the time we get to complete seawater, of course, the plants die. It's lethal. And so one of the things we did with this study was quantify the damage. So before we can say, can we alleviate the damage, we have to measure how bad the damage is with the salt, and then we can look at what we can do to alleviate it. So one of the things we decided to look at was the microbes in the soil. So this has been used for thousands, if not, I don't know, millennia, how long people have been using microbes naturally by maybe adding animal dung and compost to their crops, and noticing that this helps them be healthier and survive better. But with today's science, we know a little bit more about what's going on with the genes and the different species that are important and the mechanisms. And we can say now it's science-based that these old methods are really good. And with science, we can determine which are the best microbes to use and how best to use them. So we were looking at salt problems. So we said, let's go to an environment that is high in salinity. And we have a lot of mangrove forests around the edges of Malaysia. In fact, that area that's going to be underwater is currently mostly mangrove forest. And I know in India too, you have this kind of ecosystem. And what you notice there is, despite the fact there is high salinity, there's changing water levels due to tides. They have very high tides at equinoxes, there's high spring tides, and very high temperatures. And yet we see lots of healthy plant growth going on there. So our idea was that the microbes that are helping these plants to grow may be able to protect other plants against salinity. And this is just showing you the diversity, and you'll see that India too has a high diversity in mangrove species. And the sampling site where we went, which is just off the west coast of Peninsular Malaysia. And we sampled at different regions from the low water to the high water line, and we isolated a number of strains that were tolerant to salinity. We checked them for growth promoting activities. So these types of activity are very standard. Everybody will look for the same things. They must be able to solubilize phosphate. They must be able to fix nitrogen. They should produce siderophore to help with iron uptake, IA production to help root development, and ACC deaminase to protect against oxidative damage. And we could see that not only can these strains have these activities, when we put them on the banana roots, they are able to colonize. So even though they were not originally isolated from banana plants, they can quite happily colonize banana roots. So the experiment we did was we first of all isolated them, we cultured them, we acclimatized some plants in tissue culture, and then we treated them with the solution of these plant growth promoting rhizobacteria and challenged them against salinity stress. So in the next set of diagrams or graphs, I show you the coloring is the same and the order of the bars. So the blue bar that is always on the left-hand side is the control plants. These had no salinity, but no added bacteria. The next one is what happens to those plants when we add sea salt. So we didn't use sodium chloride. We used a mix of aquarium salts to really mimic seawater. The third column, the gray, is what happens to those plants when we inoculate them with plant growth promoting bacteria in the absence of salt. And we can see straight away that just adding the bacteria gives tremendous difference to the height and the length of the roots of these plants. So it makes them really healthy. And the last one of each set, sort of yellow color, is what happens when we add the bacteria, but we add back the salt. And what we can see is the presence of the bacteria is mitigating some of the damage, especially plant height. And even with roots, they're typically still longer than in the plants with no bacteria in the absence of salt. So this seems to be quite helpful to these plants. We can look at other indicators like chlorophyll content and proline content, sodium, potassium, electrolytes, and various antioxidant enzymes. So overall, rather than talking you through a lot of graphs, all of the data was consistent with the fact that the bananas that had been colonized with halotolerant bacteria showed better tolerance to the salinity condition. And that work was published. And so we say, yes, can stress-resilient plant growth-promoting bacteria alleviate salinity stress in banana? And the answer is yes, they can. And that's a great result. But since the plants are so healthy, we wondered, does that mean they might be more resistant to disease as well? So we had the same hypothesis that the bacteria isolated from an extreme environment will be resilient agents that actually can be used in crop protection, not just against abiotic stress, but also against biotic stress. So I mentioned earlier that one of the major pathogens for banana is a fungus called Fusarium, and the strain that we have in our part of the world is the tropical race 4, which is shown in the red dot on the map, and this is spreading rapidly across Asia. You have it in India too, and in fact there are not many countries that don't have this pest. It's extremely difficult to eradicate once it gets into the soil because the spores are very resilient. And a lot of phytosanitary treatments have to be done, but nearly all the current commercial triploid A cultivars of banana, the ones that are eaten all across America and Europe and Australia, are very susceptible to this fungus strain. And more than 80%, in fact, of global banana production is of germplasm that's susceptible to this fungus. So again, we started by getting the quantitative data of what happens when we have the fungus infecting the plant. And we have an inoculation system, which we call the double tray method. And we're using this at the moment to screen wild germplasm to look for resistance. But in the meantime, what can we do for our cultivated bananas? And what we can see is that, yes, when we infect them with this fungus, very quickly they show a lot of damage. And particularly, it gets into the vascular system, it prevents the water uptake, and it results in a severe wilting of the plants. So could the microbes that we use to alleviate against salt also alleviate against this fungus? So there are some evidence that bacteria can help, because they can antagonize with the Fusarium, and I'll show you that in a moment on the next slide, as well as promoting the growth. So they make the plant healthier, and they also directly antagonize the pathogen. So we tested some of our strains by just putting them on an agar dish together with the fungus pathogen. So the big fluffy circle is the fungus, and the line is the bacteria. So we tested 2 strains for this, or we found 2 strains that were particularly good, a Bacillus strain, which we call RB3, and a Pseudomonas strain, which we call EB3. And when we combine the 2, they actually have a stronger antimicrobial effect on the Fusarium. So now we colonized our banana plants, this time with the 2 strains. So in each case, they're colonized with both the EB3 and the RB3. And we measured them at 4 weeks and 6 weeks in soil. And so in these next graphs, again, all the samples are in the same order in each graph. So on the left-hand side, we have non-inoculated plants. So this is, they have no protective bacteria, but they're not infected with fungus. The next one, which is in the red circle in each case, is what happens when we infect them with the Fusarium. And we can see, of course, it reduces the height and the root length and biomass. It doesn't have much effect on number of leaves, but bananas don't form a lot of leaves within 4 to 6 weeks, so it's not surprising we don't see much effect. Then we add the growth-promoting bacteria, and we see everything improves, the height, the root length and biomass, and actually slightly the number of leaves. And then we add back the Fusarium. So these plants are protected with the EB3 and RB3 strains, but now they're infected with Fusarium as well. And what we see is that the damage is not as great as in the plants without the bacteria protecting them. So we are mitigating some of the damage through the presence of these plant growth promoting bacteria against this very severe fungal pathogen. And again, we can look at other things like chlorophyll, carotenoids, and antioxidant enzymes, and the story is the same, that the presence of these two bacteria together is able to mitigate or alleviate effects of the fungal infection by improving the plant health and presumably also antagonizing with the fungus in the soil. So another yes, can stress-resilient PGPR, plant growth promoting rhizobacteria, alleviate biotic stress in banana? And indeed they can. So this is very promising. It could be used as a biofertilizer and maybe a biopesticide to help protect banana plants in the field under these challenging conditions. So there are many, many studies on plant growth promoting bacteria, and I know that there's a lot of really good work going on in India in this area. Different formulations, different bacteria for different solutions, maybe for a different type of plant or a different type of soil. So there's a lot of prospects to going back to those old techniques, but using science to validate which microbes in which conditions are the best for those crops. So now I'm going to move on to another study, and this is the use of a natural gene regulatory system based on small RNAs to produce RNA spray pesticides and biomodulators for crops. So I'll start with a bit of background on the small RNAs. Plants naturally have microRNAs, and they also have another system alerting small interfering RNAs or siRNAs that carry out this process of RNA interference, RNAi. It's also known as RNA silencing. So you'll read different papers, you'll see different terms, and basically they are the same process. It is a sequence-specific silencing, so we use the complementary strand to the RNA that we want to prevent from being translated. And that means we can very precisely target a messenger RNA so long as we know its sequence. Now, it is a newish tool, but it's again, biology invented it, not humans. Humans are just clever enough to discover how to use it. And the very first ones were based on antisense. But one of my heroes in the world of RNA silencing is Sir David Bolcombe, who's from UK. And he wrote a review and also credited, he said, this is new science, but not really. If you look back in the literature, here is a paper from 1928. So we're going back almost 100 years. And this study describes how when you had a plant that was infected with a mild strain of the virus, the plant was able to recover. And if you try to reinfect it, it didn't get infected. It had somehow gained an immunity to that mild strain of virus. And so we could apply milder strains of virus and protect crops against severe strains of virus, provided they were using the same RNAs. That wasn't known at that time, but now we know that's why it works. And the mechanism was better understood, later refined as double-stranded RNA silencing, and they were some of the first crops used in breeding. Actually, the very first genetically modified plant used something called antisense RNA. Before the double-stranded RNA mechanism was really understood, it was hypothesized that it should work because if we have a messenger RNA, it's single-stranded, and the ribosome needs to see that single strand in order to make protein. So if we have an antisense, a perfectly complementary RNA added in, it will bind through hydrogen bonding and form a double-stranded RNA, which cannot be translated by the ribosome because it can't get to the molecule it needs to translate. So by adding an antisense, we could sequentially, specifically silence any messenger RNA. And this was the principle of antisense RNA. And that was used to make the Flavor Saver tomato, which was the first genetically modified crop to be commercialized back in 1994 in the USA. It didn't last long on the shelves, but it was commercially available in both US and Europe for a little while. And also, as well as targeting genes within the plant, so in this case, they were silencing polygalacturonidase, which causes the fruit to over-ripen or become very soft and susceptible to spoilage. We can also target viruses, like in this case, papaya ring spot virus. So the plant on the left-hand side is transgenic. It's expressing antisense to the viral RNA. And the one on the right-hand side doesn't have that protection, and you can see it's severely damaged by the virus. Later on, it was actually found that microRNAs and other types of double-stranded RNAs, such as from viruses within cells, are triggering an RNase enzyme to take them up, cut them into small pieces, and then look for matching RNAs and cut those into pieces or block them from translation. There's also a pathway actually that causes them to be methylated and can cause DNA to be silenced, but we're not talking about that today. We're focusing on the RNA, and by cutting up the RNA, it's not available to be made into protein, and we can reduce the amount that is made. And that was actually an accidental discovery. Fire and Mello, who won the Nobel Prize for this discovery in C. elegans in nematode worms, actually discovered it as one of their control experiments where they put in double-stranded RNA. So always be looking for those weird results. Don't throw them away. Think about what is it telling you, and you might just get a Nobel Prize someday like they did. Okay, so we can actually put in genes into plants to make this antisense or to express these double-stranded RNAs that trigger the silencing. The problem with making genetically modified crops is that it's quite expensive. It takes a long time. You have to have a transformation and regeneration system for your plant of interest, and that's not always easy to do. In fact, I was just discussing last week with someone, they still haven't managed to do it for coconut. So there's still plants where nobody can do it yet. And of course, there is some public perception and concern with transgenic crops. So what if instead of making the plants make the double-stranded RNA molecules, could we just put double-stranded RNA onto plants and cause gene silencing this way? And this is what this clever group back down in 2003 in Spain did. They made double-stranded RNA using bacteria against a virus. This was a plumpox virus. And they treated one side of the leaf with the double-stranded RNA and the other with the control RNA. And then they infected with virus. And they saw that the double-stranded RNA was able to protect the plant against the virus infection. So it was actually interfering. And it was just by putting the RNA on the leaf, not actually making the RNA inside the plant. So this is not transgenic. We call it non-transformative RNA silencing or RNA sprays. Can we just spray the RNA onto the leaf and protect it against the virus? So we can remember that it's nucleotide specific. It is triggered by double-stranded RNA in a hairpin or as a double-stranded RNA, so we can choose different ways of adding it in. And it doesn't get into the nucleus. So it's a transient effect. It's not a transgenic plant, but it does have a genetic effect where it silences that specific gene. So we saw this and thought, that's really cool. I want to try. And what should we do? So we said, well, what plants in Malaysia have a virus problem? And we came across orchids. Of course, they are beautiful, and there's quite a big floriculture in Malaysia and Singapore. Actually, Singapore's farms are usually in Malaysia, but they're owned by Singaporeans. And it's big business. And you can see that if a flower is infected, it becomes very ugly and impossible to sell. And this Cymbidium mosaic virus is very, very common, and it infects all kinds of orchids. So we wanted to know, could we spray our plants and protect them against this virus? So here was our scheme. We will take a double-stranded RNA, which is targeting the coat protein. So Cymbidium mosaic virus is a Tobomo type virus. So it has protein coat and an RNA core. The RNA becomes a messenger RNA. And if we make a complementary double-stranded RNA, this should target it to be degraded and prevent the coat protein from being made. And if the coat protein isn't made, the virus can't assemble and spread inside the plant. So this was our first work in this area, and we decided to do all the controls, to do a sense single-stranded RNA and an antisense single-stranded RNA, as well as mixing those together to make a double-stranded RNA and see if they are able to protect the plants. So what we did was we treated the plants, infected them with virus, and then we tried to detect the coat protein RNA by using PCR. So this is a quantitative PCR of the amount of virus coat protein RNA in the samples. So in the lane 1, we have the positive control. The plants were infected with the virus and not treated with any RNA. So this is what normally happens. We get lots of virus, the virus multiplies, and that is the result you get. When you treat them with the antisense, that is in lane 2, or the sense, that is in lane 3, we can reduce the amount of virus that you see. Now, when we treated with the double-stranded, we got effectively a 0 reading, and I'm putting up the true reading, so you know they're noisy. It's not really, cannot be less than 0, but you can get this kind of result when you do quantitative PCR. And in lane 5 is the result for plants that were not treated with virus or any RNA, and in lane 6 is the plants that were treated with double-stranded RNA and not infected with virus. So we could see that in the presence of the double-stranded RNA, it protects completely against the virus. And this is just by spraying it onto the plants. So we have gone on to patent that, and the work has been published and won some awards. Next, more recently, we found a local nursery that was having a problem with cucumber. And this is infected by cucumber mosaic, melon. It's cucumber mosaic virus, but melon is in the cucumber family. And the melon farmer was having virus problems and asked us, could we work with them? And they were interested in collaborating. We managed to get a little grant to do this. So we first of all determined what the virus was, and we found it was a cucumber mosaic virus. And this time we decided to use both the coat protein and the movement protein genes to see if we could protect the melons from the virus using a spray. So this is a summary visually. Of course, we did more than one plant each, but just to show you what the leaves look like when they're infected with virus. So the red box is showing you at 10, 20, and 30 days after inoculation with the virus, what the symptoms normally look like on the leaf. And at the bottom, if we don't infect with any virus, this is what the healthy leaf should look like. So on the top, this was treated with double-stranded RNA to the coat protein. And the next one was treated with double-stranded RNA to the movement protein. And the next one in the middle was treated with both coat protein and movement protein double-stranded RNA. And we can see that when we treated with both the RNAs, we got pretty good level of protection. But even with each of them, we got some level of protection. There's less symptoms than without any double-stranded RNA treatment. So we can then actually use ELISA as well as our visual symptoms and come up with what we call a disease severity index score. And again, the blue is with the coat protein in each case, the red is with the movement protein, and the gray is with both, compared to the levels of virus when there's no RNA used. So we get a good level of protection, and it's strongest when we use more than one targeting gene. So that's very exciting. And so this was also published, and I'm just going to read you the title because we did a little bit more in the paper. We said control of the cucumber mosaic virus in rock melon using double-stranded RNA-mediated silencing of coat protein and movement protein with no deleterious effect on plant phenotype. So one of the questions we often asked is, what happens though if you start spraying it with this RNA? Maybe it affects other genes in the plant and you get what we call off-target effects. Or what about if it affects one of the pollinating insects and you get this effect on the pollinator? Does it affect the honeybees or so on? So actually in this paper as well, we described that we checked using bioinformatic techniques that there was no impact on other genes in the plant and also no impact on honeybees for this plant. So we were able to show that it's actually quite a safe technology to use. Now, another thing we were interested in, though, since we can not only target viruses, but if you remember the earliest antisense on polygalacturonidase, we can actually alter plant genes themselves. can we do anything like modifying flowers? So in this work, we actually went back to orchids and we tried silencing one of the transcription factors, which is an R2-R3 MIB gene. And we found that although our original hope was we could maybe change the shape or color of the flower, we couldn't, but we actually did change the shape of the cells on this part of the lip of the orchid, which was visible under electron microscopy. And what we can say from this is, one, we can make such a change, and two, we can discover what the gene is doing, because now we know that this particular gene, if we treat it onto the orchid cells, is very specific to affecting the lip epidermal cells and changing their shape. So this can actually be an important tool for determining what a function of a gene is. And we don't have to do any transformation. We just have to spray double-stranded RNA onto our plant and see what happens. So it's much faster and cheaper than doing transgenic or knockout studies. What about really changing the shape and color? Well, we kept going. We managed to get another grant so we could do some more genes. And in this one, again, the red box shows you what the normal flower development would be like, either with no RNA treatment or treated with an empty vector. So this is the shape and color of this Dendrobium flower with no RNA treatment. And in the three above, we had double-stranded RNA to another transcription factor MIB3, in the middle, transcription factor MIB4, and actually to the chalcone synthase, which is one of the anthocyanin pigment biosynthetic pathway genes. And what we could see with these was that we can get a paler color, which we expected with chalcone synthase, and we hoped with the MIB genes, and we did, but we also seemed to change the flower shape. So this was something new. And how do we quantify something like that? I can tell you one flower and I maybe picked the palest flower, so that's not very good. I need to do lots of flowers and to quantify so we can get some statistics. So we can actually do color change by measuring something called delta E, using a colorimeter of lots of flowers, and then we can actually show a statistically significant difference in the petals, in the sepals, and in the lips, using these different double-stranded RNA constructs. And what about shape? We can see it, but how do we quantify it? So in this case, I challenged my student to find a way to quantify it, and he did. I was very pleased with him. He came up with a way of mapping coordinates of the photographs of all the flowers, and then we can superimpose them and measure the shape distance for the sepals and the petals and also the lip. And then we can do lots of flowers and we can get statistics again to prove that it is quantitatively different and statistically valid. And we found that treating with these different transcription factor double-stranded RNAs actually do quantitatively and statistically significantly change the shape and change the color of flowers. Now, what was interesting about this was that before this research, these transcription factors had been kind of associated with flower development, but they didn't think the same transcription factor could affect both color and shape. And we showed that they did. So it was actually good functional study as well to show that one transcription factor may be doing more than one thing, at least in the flowers of orchids. So that was a very nice study. So that's the end of my research or our group's research. But what I want to tell you is, where is it going? Because you're still young people and you want to say, like, is this something that's real, or it's just like in the lab? It's just something showing that these genes do this and that, but it's not going to be useful to farmers. It's too difficult. Well, in 2024, the first RNA sprays were actually commercialized. So they are actually out there in the market. And this one is really nice because we were doing viruses and we were doing, genes within the plant. This one actually targets a beetle. So it's very, very different target, the Colorado beetle, which is a real pest of potato plants. And there's a company in the US, you can look them up, Greenlight Biosciences, and their product is called Calantha, and it is an RNA pesticide. And it just took them 4 years. Okay, 4 years seems like a long time. That's actually pretty fast to get a biotech product into the market. Okay, so they then have been working on other things. They are now actually working on a RNA spray against a mite that infects honeybees. Okay, and this is an interesting example because someone did a study to say, what do farmers think of that? You know, the mites are affecting our honeybees, but are you ready to use an RNA spray to control that? And in fact, they found, this is a study in New Zealand, and New Zealand doesn't like GMO. They don't grow GMO. They don't like to eat GMO. But the beekeepers in New Zealand said, yes, we like this technology. It's going to protect our bees. And it's not GMO because it's just a transient expression of double-stranded RNA, and they're happy to use it. So this, to me, is really encouraging of how we can, even where people are resistant to some of the technology, they're still not resistant to all technology, especially if they can see how it will benefit them. So this is a really happy story. So to summarize what I've told you, the mangrove forest is very useful. We can find all sorts of stress-tolerant microbes, and you have your own forests over there. So I'm sure you have tons of interesting microbes that are just waiting to be used. When we use these halo-tolerant bacterial isolates, we can have multiple plant growth promoting activities. They are able to colonize bananas and likely other crops. And those bananas are tolerant of salinity and have improved tolerance to fusarium, which is one of the major plant pathogens for banana. I also showed you that RNA sprays use naturally occurring gene silencing that happens in many multicellular organisms, and we can protect against viruses. And we don't need to have a GM plant, so it's much faster. So imagine you have a new virus emerging. You don't have to wait for the transgenic variety. You can just make a spray and straight away protect your plant against that new virus. So it's going to be really good for outbreaks of new viruses in crops. They also can modulate other commercially valuable traits, including flower shape and flower color, and probably flavor and all sorts of other things. So we haven't really gone deep into what we could modify that could be of value to farmers. So adding biotech tools to best agricultural practices to address food security can improve the value for farmers and can be a start of a really good biotech industry. Excuse me. I need to cough, so I'll just put up all the people I want to thank. And you can find our publications online. And thank you very much.
Thank you so much, Professor Jennifer, for that insightful and inspiring talk. You have given us a deeper understanding of how biotech tools for sustainable topical crop production can enhance resilience to climatic change, improve crop yields, and promote environmentally friendly agricultural practices. We will now proceed to the interactive question answer session. Participants, please post your questions in the chat box or use raise hand option if you would like to speak. While we wait for your questions, I will begin with a few to get us started. My first question is, Professor Jennifer, could you please highlight the most promising biotech tools currently being applied to improve the sustainability of topical crop production?
Thank you, Divani, for the question. I think that tropical crops are facing the same problems of all crops. We have competition for land and water. We have climate change, and we have pests and diseases. And so the tools that I've shown you are some of the tools that can help. We can also use marker-assisted breeding, gene transfer. GMO, I believe, is still a valuable tool, even though people are sometimes resistant to it. But what is promising now is that gene editing seems to be more acceptable to many people, and that gives us a lot of possibilities to improve traits that can cope with lower imports or climate challenges and so on. And the other thing I want to say is that biotech will actually benefit from working alongside other types of technology. So AI and automation technologies are going to be really important for food security in agriculture. And they can be important tools for helping you actually do better biotech. So for example, if you want to make a new RNA spray, our AI tools may be able to predict the best targets for you and things like that. So we need to work with many different technologies. Another work that I didn't talk about here is that we have worked with lighting engineers to look at growing crops indoors. So one of the ways you can reduce some of the impacts of climate change is to produce some of your food, not all of your food indoors, but some of your food indoors, especially small crops, fast growing and highly valuable. And we can actually work with different wavelengths of light. But in the future, we can maybe modify the plant to work better with different wavelengths of light as well. So this will be something using artificial light. So I think it's a very exciting time to be combining all the technologies and looking for lots of different solutions and not limiting yourself of what can be used.
Thank you for sharing your insights on how these tools are shaping the future of agriculture. My next question is, in your case studies from University Malaya, what specific challenges of tropical agriculture did biotechnology help to overcome?
Okay, so we also do quite a lot of work on the genes and genomes, transcriptomes, and I think one of the challenges is getting the funding and also looking after your data, right? Because you may be a bioinformatician, but you may not be, and you may not consider that managing the data and storing the data. We just, I'll give you a very painful real example. Our server just crashed, right? And we may have lost a lot of data. And this is a very real problem for a lot of universities, especially in developing countries. So this is something that I would say is a big challenge to us. Finding funding to do the research and to maintain the facilities, and also sometimes find the people, and also the people who we can afford. If we can't pay big salaries, but we need more bioinformaticians, for example. So I think Malaysia, India, we see a big brain drain. So this is also one of our challenges. You know, how do we grow the industry so that the salaries are fair and that we can keep our good people to work with us.
Thank you for illustrating how research is directly addressing the real-world agricultural issues. Next question is, how do molecular genetics approaches complement traditional breeding methods in enriching crop resilience under tropical conditions?
Okay, thank you for the question. So I think traditional breeding is still really important. In fact, the meeting I just came back from in Banana is aimed at doing traditional breeding. But the molecular genetics can really help us identify what the genes are doing. So for example, breeders traditionally can focus on traits they can measure, but they can't always know what all the genes are doing. Like the transcription factors I just described, we didn't know that they actually have a big impact on shape and color. These are important phenotypes. So this is where the molecular genetics is very powerful. Gene editing can make a change more quickly than breeding. If it is a single, or soon we'll be able to do multiple genes, and then it may be faster to develop the variety that you want and avoid traditional genetic engineering. So I think there's a lot of ways that biotech tools and molecular genetics really can work together with traditional breeding. Also, traditional approaches like using the microbes in the soil, we now know that they work, but now the molecular genetics can tell you, you can go and look for a particular enzyme in a particular microbe that is going to be helpful. So these all can really enhance, not always replace, but definitely enhance the traditional technologies.
Thank you for explaining how modern and conventional approaches can work hand in hand for sustainable solutions. Hello participants, do you have any questions? If yes, please unmute yourself and ask your question. All right, I see some questions in the chat box from Dr. Ashik Narjay. Professor Jennifer, we will best quality of prawns, sustables and Kashmir, India, but with low productivity. Could you tell us how can we increase productivity?
Okay, this is very tricky because particular crops have their own issues, and I think the Crocus sativus is this the one used for saffron? I think it's a very I think it's not a easy plant to work with. So normally, one of the ways people can boost is a smallish plant, so perhaps it's something that can benefit from tissue culture techniques to multiply. I think, like all plants, we can look at the growth-promoting bacteria to see if there's any particular bacteria that can help it grow faster or healthier. But sometimes there is not much you can do to improve a crop if you're working with a single species. I'm not sure how much breeding has been done in crocus, so I'm sure people are looking into that as well.
Thank you.
Thank you
Thank you.
for addressing. Thank you for addressing the question so clearly. Your answer have given a valuable insight into the topic. Now, next question is Prabhat Kumar. It is possible if you will come use mathematics parts for microfigures, vegetative tissues, virus infection will be less.
Yes, so this is a good technique for propagating plants. If you have a plant that is easy or not easy necessarily, but it's possible to do micropropagation, you can get very clean planting materials. But the issue is then keeping them free of virus as they go on. So they will start virus-free. But if you are growing them where they get exposed, so I'll give you the example of orchids. The orchid virus actually is not carried by an insect, but it is in the sap of the infected plants. So if you have a plant and you don't know it's infected, and then you have sap on your gloves and you're handling other plants, it's very easy to transfer it. That's why it's spread very easily through the sap. So once it's infected, you can't do much. But if you knew you're going to handle the plant and you spray it with the RNA spray, you can protect it during the handling. So it's fine if you don't handle it, but as soon as you have to handle it, you may need to protect the plant. And so this is where there is a place for the spray. But definitely for the micropropagation and multiplication, that you need to start with clean planting materials, and meristematic tissue is one of the best ways.
We thank you. We thank you for that explanation. The next question is from Dr. Dev Singh Thakur. For Indian farmers, high productivity is major criticized in crop production. Does sustainable crops production develop focuses on these or support in these?
Yeah, this is a very good question. When we look at what a lot of people are saying today is that when we go to very high intensive productivity, we tend to do a lot of things that are not really sustainable. You know, we will use a lot of agrochemicals. We will push the plants very hard to multiply. We use monoculture, so we're reversing the biodiversity in an environment. So they seem to be opposed to each other, particularly if you're talking about environmental sustainability. So this is where I think we need to get back some balance. It's impossible to do agriculture without disturbing biodiversity. It's going to happen. But can we minimize the impact? So if we use less of the harsh agrochemicals and more natural fertilizers, compost, and biofertilizers, biopesticides, we can reduce the impact, yet still maintain high productivity. So I think we have to find a balance. I see people now saying that we should intercrop more, so you don't just grow monocrops, maybe grow a few different varieties at the same time, and so on, but still balance it so that you have high enough productivity. And this is another one where I think AI and machine learning models can come in. They may be able to tell you which are the best crops to grow together and when, and which genotypes can be mixed and work together, so that you can still get that productivity, but you can have like maybe better biodiversity and less environmental impact from the agriculture. But it's impossible to have a zero environmental impact. Agriculture disturbs the environment. There's no way we can avoid that, but we can try to minimize that.
Thank you for patiently answering the questions. Your input has added great value to our understanding. Next question is, could you share an example of a successful collaboration between academia, industry, and farmers that accelerate the application of biotech solutions in topical crops?
Okay, so here I'm going to go away from crops, because from our center, we have done a lot of more upstream work. It hasn't really reached the farmers. We get as far as patenting, but we had not developed anything that is gone to farmers. But I have colleagues in our center who work on aquaculture, and they have developed some diagnostic test kits for viruses, for shrimp and fish. And those have been commercialized and are being used by farmers. So there's an example of something that really has reached to the farmer. I think it takes a long time to get a product, and it's not always easy for university researchers to focus on that part of it. So if we are lucky, we will meet with companies who are interested to do that development and commercialization, because I think it's quite hard for us to do it ourselves, and maybe it's not so exciting for us. We like to just do the lab research. We're not really farmers ourselves. So we need to work with partners and colleagues in order to get those research translated. But it is very important that research does get translated. So I think the best way to do it, if you're not going to do it yourself, is to try hard to network with people who are interested to do that development.
Thank you for showing how partnership can speed up innovations and create real impact for farming communities. Next question is, what are the major biosafety and regulatory considerations when deploying biotech-based innovations in topical agriculture?
Yeah, so in terms of biosafety and biosecurity, there are two aspects. One is the genetic modification, and the other is just the pests and diseases. And they're very important, both of them. So the regulations are different in different countries. It is good if we can see more international harmonization, which will allow us all to agree. It's interesting, though, for gene editing, many countries seem to be adopting the same new rules, which is that they accept very small deletions or very small additions as non-GMO products, which will be much easier to get into market. Malaysia has also done that. I believe India is also taking that approach. So I think that some of the barriers are going away, particularly with the newer technologies. RNA sprays, as you've seen, is already commercial in USA. So I think you will see better uptake. But each country will have to decide based on a combination of their own policies and their own population, because policy isn't science. It should be informed by science, but it is still up to people to accept whether they're going to take these new products and feel comfortable with them. But I think over time, it will definitely get better. It's getting better all the time.
Thank you for clarifying how safety and regulations guide the responsible uses of biotechnology. Next question is, how can students and early career researchers contribute meaningfully to advancing biotech applications for sustainable tropical agriculture?
Okay. So if you are doing any kind of research in a small project or an attachment, you are contributing already, because the human factor is still important. I mentioned AI and all these things, but we still need people, and people are the most creative. So I think I would say you need to be very open-minded. Look and learn as much as you can about biology, because like I said today, many of these things that are being used already exist in biology. It was just somebody seeing how it could be applied for biotech. So that's one. I would say also you do need nowadays to familiarize yourself with AI and other technologies, because they are going to be with us, whatever happens. So you need to know how to use those technologies to make your life easier and be better informed. And the third thing that I think every young researcher needs to know is how to write a convincing proposal. Because one of the very big challenges everybody faces is getting funding. So whether that is for your scholarship or a postdoc or whatever project you're going to do, when you're seeking funding, you're always going to be competing with other people, and everybody thinks their idea is the best idea. But you have to make sure when you write your proposal, your idea looks like it really is the best idea. And one of the ways to do that is to really address what is the problem you're going to solve, and who says it's a problem. If your funding body has listed that these certain SDGs need to be addressed, like food security, and you can say, oh, my proposal will help address food security, then you're already partway to convincing them to take your proposal seriously. So it'd be great to just do what I like, but we have to sometimes do what they want. So we have to match what we like and what they want, and then we get a good proposal.
Thank you for encouraging the next generation of scientists to play a role in these important fields. Looking ahead, my last question is, what future direction or emerging tools is in biotechnology excite you the most for improving food security in tropical regions?
Okay, so I've talked about some of the ones that we've been doing, and they excite me. I'm excited about using RNA sprays. I'm excited about gene editing. We also have some gene editing projects going on. I'm very excited about the potential for microbes. So I will just talk two topics. The first one is with RNA. I mentioned that we can actually modify DNA as well. We can change the epigenome, but nobody has a tool for doing that reliably yet. So I think that's going to be another big breakthrough. Just like RNA silencing, if we can controllably and predictably modify the epigenome, and people are working that in medicine, for example. But can we do that for crops? I think this is going to be a very useful tool, and that will be passed on through more generations, but not necessarily needing you to do genetic engineering directly. So that's one. So I challenge someone to do that, because I think if they can crack it, that could be a Nobel Prize for someone. Okay, and the second area is microbiome. Okay, so I've talked about adding 1 or 2 strains, but actually now with the amount of information we can get and the tools to manipulate and analyze the data, we could be doing whole host complex mixtures of microbes. And just like they've been shown in human health to be really important, I think in plant health, they're really important. So there's still a lot of discoveries to be made in plant microbiome research. And I think this is another very exciting topic for the future. Thank you.
Thank you for sharing your vision of the future, and this is responsible for holding for agriculture. All right, fellow students and esteemed participants, that concluded with a truly enriched discussion. I take the opportunity to thank Professor Jennifer for sharing her profound insights and expertise with us today. To all our participants, thank you for your active engagement. With that, I now hand the session back to our MC. Thank you. Jack, over to you.
Thank you, Devrani, for moderating the session. As we draw this exciting session to a close, I now invite Ms. Anushka Yukri to deliver the vote of thanks on behalf of the entire organizing team. Anushka, over to you.
Thank you, Sahib. A very warm good evening to everyone. My name is Anushka Oike, and I am student of BSc Right Technology People's University, Bhopal. As we draw this insightful session of the BioConnect webinar series to a close, it is my privilege on behalf of the entire CSRB student team at People's University to deliver the vote of thanks. A special thanks to distinguished guest speaker, Professor Dr. Jennifer Harikrishna, for delivering a thought-provoking talk on biotech tools of sustainable topical drugs production. Case study from University Malaya, Professor Jennifer. Your insights have broadened your academic perspective and ignited your curiosity to explore real-world solutions through biotechnology. We extend our deep appreciation to our mentor and guiding light, Professor Dr. Subhash J. Bharat, head of CSRD, for his unwavering support and encouragement of students led initiative like this one. So your vision and leadership continue to inspire us to aim high. A heartfelt gratitude, a heartfelt thank to our register moderator, Ms. Devrani, for smoothly steering the session and encouraging meaningful dialogue. We also sincerely thank our organizing committee chairperson, Mr. Mohammad Ashan, the committee member, and the entire CSRG student team. Your dedication and behind-the-scenes efforts has been instrumental in making today's event success. To all your enthusiastic participants and attendees, thank you. Your presence and engagement bring life to this platform and motivate us to grow and learning together. This is just a beginning. The BioConnect webinar series in enriching has widened students-led platform where ideas, abilities involve, where students evolve into lead, and where knowledge translates into meaningful action. We hope today's session was just enriching for you. It was for us, and we look forward to welcoming you to the future session of the BioConnect webinar series. For the student by the student, before we formally conclude, we would like to record a special thanks to your management of People's University for this continued support, which remains the cornerstone of academic excellence at your university. Esteemed participants, thank you once again for being with us today. May your journey be filled with curiosity, innovation, and meaningful discovery. Wishing you all a pleasant evening. Now I will hand it back to your MC, Ms. Zahid. Ms. Zahid, over to you. Thank you.
Thank you, Anushka, for expressing our sincere gratitude to all respected guests, our expert speakers, and every participant. Fellow students and esteemed participants, Before we signing off, we request all participants to kindly take 2 to 3 minutes to fill out the post-webinar feedback form. The feedback form link is posted in the chat box. Your response are extremely valuable and will help us make the future events even better. We will wait 2 to 3 minutes to let you complete the feedback form. We hope you have completed the feedback. Please note that your e-certificates will be sent to your registered email ID shortly. We sincerely thank you once again for being part of this encouraging session. We look forward to welcoming you to our next BioConnect webinar very soon. Until then, stay curious, stay inspired, and keep connecting. This is Chehek Soni signing off on behalf of the entire organizing team, People's University, Bhopal. Have a wonderful evening.